About the peripheral nervous system. The peripheral nervous system is going to provide our link from the outside world, including outside of our central nervous system in our body, and the outside world, literally outside our body. Communication between that and the central nervous system. It's got neural structures outside the brain and spinal cord, and we're going to look at breaking them down into four major parts. The sensory receptors, the transmission lines themselves, which would be the nerves and the structure, the motor endings, motor activity, and then we'll look at reflex activity. So we've talked about the central nervous system. We did that last week. This week you will be dissecting a brain as part of the lab. But we're outside the central nervous system discussing the peripheral nervous system, which includes the sensory division and a motor division. Remember, the motor division is divided into the somatic and the autonomic nervous system. Today we're going to be talking mostly about somatic. Next week we'll finish up the semester, looks like, with the autonomic nervous system. And that autonomic nervous system is divided into sympathetic and parasympathetic divisions. So let's start with the sensory receptors and sensation. Sensory receptors are specialized to respond to changes in the environment. We call those stimuli. And that activation results in graded potentials that trigger nerve impulses. Awareness of the stimulus is what we're going to call sensation, and interpretation of the meaning of the stimulus we'll call perception. Both occur within the brain. But there are three ways we can classify receptors, either by the type of stimulus they respond to, their location in the body, or their structural complexity. So if we look first at stimulus type, we have mechanoreceptors that respond to mechanical changes, things like touch, pressure, vibration, and stretch. Thermoreceptors will be sensitive to changes in temperature. Photoreceptors respond to light energy. And chemoreceptors respond to chemicals that are dissolved, in most cases, in body fluids, things like taste, smell, as well as changes in blood chemistry. Nociceptors are sensitive to pain-causing stimuli, so extremes in heat or cold, excessive pressure, those things will trigger nociceptors. Exoreceptors are going to respond to stimuli that comes from outside the body. So the receptors we have in our skin for touch, pressure, pain, temperature, most of our special sense organs, that's another class of things we'll look at next semester. Interoreceptors or visceroreceptors are going to respond to stimuli within the body from our internal viscera and blood vessel. They're sensitive to things like chemical changes, stretch, and temperature. Sometimes they cause discomfort, but usually we're unaware of their workings unless they're at extremes. Proprioceptors are going to respond to stretch in skeletal muscles, tendon, joints, ligaments, and connective tissue, and they are helping to inform our central nervous system of movements and position of our body at any given time. So the majority of receptors belong to either simple or receptors for special senses. The simple receptors are modified dendritic endings of sensory neurons. They're found throughout the body, monitor most types of general sensory information, while the receptors for the special senses are all found within our head, and they are for vision, hearing, equilibrium, smell, and taste. They're housed in complex sense organs, and we will be covering those at a later chapter. The simple receptors for general senses include tactile sensations such as touch, pressure, stretch, and vibration. There's no one receptor, one function relationship. These general senses can respond to multiple different stimuli. The receptors themselves can have non-encapsulated or free nerve endings, or they can have encapsulated nerve endings. The free nerve endings, non-encapsulated, are abundant in our epithelia and connective tissue. Most of these are non-myelinated, small diameter group C fibers. Their distal terminals have a knob-like swelling. They're going to respond to things such as temperature, pain, or light touch. Thermoreceptors come in two different kinds. There are cold receptors and heat receptors. The cold receptors are activated by temperatures between 10 and 40 degrees Celsius. Remember, this is Celsius if you're used to Fahrenheit. They're located in the superficial dermis. The heat receptors are activated between 32 and 48 degrees Celsius, and they are located in the deeper dermis. Just for reference, 37 is normal body temperature, 37 degrees Celsius. Outside those temperatures, nociceptors or pain receptors are activated, and that would be interpreted as pain. So those nociceptor pain receptors are triggered by extremes in temperature, pinch, or release of chemicals from damaged tissue. Some of them would include the vanilloid receptor, which is a protein in the nerve membrane. It acts as an ion channel that's opened by heat, low pH, and chemicals. Capsaicin is a chemical that interacts with the vanilloid receptor. There are also itch receptors in the dermis. They can be triggered by chemicals such as histamine. The Merkel's discs or tactile discs function as light touch receptors. They're found in the deeper layers of the epidermis. There are hair follicle receptors. They're free nerve endings that wrap around hair follicles. They act like light touch receptors. They detect the bending of hairs, so it allows you to feel insects as they land on your skin, mosquitoes, for example. And they're in figure 13.1, there's a very nice figure showing each of these, which you should go through on your own. The encapsulated dendritic endings are most of the mechanoreceptors, whose in their terminal endings are encased in a connective tissue capsule. They vary greatly in size and shape. We have tactile, Meissner's corpuscles, which are small receptors involved in discriminative touch, found just below the skin, mostly in sensitive and hairless areas like our fingertips. Lamellar, traditionally referred to as Pacinian corpuscles, are large receptors that respond to deep pressure and vibration when first applied, and then they turn off. They become sensitized. Those are located in the deep dermis. We have Ruffini's endings, or bulbous corpuscles, which respond to deep and continuous pressure. They're in the dermis. Muscle spindles are spindle-shaped proprioreceptors responding to muscle stretch. The tendon organ is a group of proprioreceptors located in tendons, and they detect stretch. Joint kinesthetic receptors are proprioreceptors that monitor our joint position and motion. And again, these are shown in figure 13.1. Processing. In order for us to survive, we have to both be able to detect changes in the external environment and consciously interpret what those stimuli mean. So we have sensation, which is the awareness of changes in the internal and external environment, and perception, which is our conscious interpretation of those stimuli. So we have a somatosensory system, which is part of the sensory system serving the body walls and limbs. It's going to receive input from exteroreceptors. Remember, those get information from outside the body. Proprioreceptors, which get information from our skeletal muscles and tendons. And interoreceptors, which get information from our visceral organs. All that input is relayed toward the head, towards the central nervous system, but there is processing that happens along the way. We have neural integration in sensory systems at the receptor level, the circuit level, and the perceptual level. So at the receptor level, we have sensory receptors. At the circuit level, we have processing in the ascending pathway, so as the information is making its way up to the central nervous system, it's being processed. And then in the perceptual level, we have the cortical sensory areas of the brain that figure out what it means. So we generate a signal. So first thing that has to happen for any sensation to occur is the stimulus must excite a receptor. that must create an action potential that reaches the central nervous system. So the stimulus energy has to match the receptor specificity. So touch receptors don't respond to light energy. Light energy doesn't respond to sound. The stimulus has to be applied within a receptive field, an area where one of these receptors are. And it must lead to transduction. The energy of the stimulus must be converted from a graded potential or generated potential and or receptor potential, depending on which one you're talking about. But that graded potential has to reach the threshold to become an action potential. Otherwise, we won't sense it. Adaptation is a change in sensitivity in the response to a continuous stimulus. Receptor membranes will become less responsive. Receptor potentials will decline in frequency or stop. We do have some phasic receptors that are fast adapting. They're going to send signals at the beginning or the end of a stimulus, so pressure, touch, and smell are kind of like that. When we have tonic receptors that adapt solely or not at all, so nociceptors, most proprioceptors don't adapt. At the circuit level, pathways of 3 neurons conduct sensory impulses and receive from receptors upward to appropriate cortical regions, and then we'll give these first order, second order, and third order names. So first order sensory neurons take impulses from the receptor level to spinal reflexes or to second order neurons within the central nervous system. A second order neuron will pick it up there and transmit the impulses to third order sensory neurons. Those third order sensory neurons will take that information that conduct that impulse from the thalamus to the somatosensory cortex. This is where we get our perceptual level. And that perceptual level is the interpretation of sensory input, which depends on the specific location of target neurons in the sensory cortex. So we have some different aspects to sensory perception. Perceptual detection, the ability to detect a stimulus. That's going to require a summation of impulses. We can also make a magnitude estimation, intensity coded in the frequency of impulses. Spatial discrimination, we can identify sites or patterns of stimuli that we can look at in the lab with a 2-point discrimination test. We have feature abstraction, which identifies one or more complex aspects of several stimuli properties. Quality discrimination, submodalities, is it a sweet taste or a sour taste? And very important, pattern recognition. Is this a familiar, significant pattern in stimuli? So pain perception is very important. It warns of actual or impending tissue damage, and so that you can take preventive action, protective action. In general, when something hurts, you should stop doing it. The stimuli for this include extreme pressure and temperature, chemicals such as histamine, potassium, ATP, acids, and bradykinin. These impulses are going to travel on fibers that release neurotransmitters, and they tend to be glutamate and substance P. Although some pain impulses are blocked by inhibitory endogenous opioids, endorphins. We all perceive pain at same stimulus intensity, but our tolerance to pain does vary between individuals. So what we refer to as sensitive to pain means a low pain tolerance, not a low pain threshold. There are genes that help determine pain tolerance as well as response to pain medications. And there's research going on in the use of genetics to determine which drugs, which pain treatment is best. Visceral and referred pain. Visceral refers to stimulation from our visceral organs or receptors, vague, aching, gnawing, or burning. These can be activated by tissue stretching, ischemia, chemicals, muscle spasms. The key here is that we generally don't get information, conscious information, from these visceral receptors. So it can lead to something called referred pain. pain from one body region that's perceived by us as coming from a different region. It turns out that the visceral and somatic pain fibers travel along the same nerves, so the brain is going to assume that they're coming from a common somatic region, which is one of the reasons that heart pain is often felt in the left arm. And there are some of these that we know about. And as you get into your clinical practice, you'll become aware of them as well. So long-lasting or intense pain, such as limb amputation, can lead to hyperalgesia, which is pain amplification, chronic pain, and phantom limb pain. NMDA receptors activated by long-lasting or intense pain. They can also learn, the spinal cord can learn hyperaglesia, and early pain management could be critical to preventing that. That phantom limb pain is when pain is felt in a limb that's been amputated. And now we use epidural anesthesia during surgery to reduce phantom pain. The transmission lines, the nerves, their structure, and their repair. Starting with nerves and associated ganglia. So the nerve is a cord-like organ of the peripheral nervous system. We've talked about this before. It's a bundle of myelinated, non-myelinated peripheral axons enclosed by connective tissue. They come either off the brain or the spinal cord, so we have spinal or cranial nerves, depending on where they originate. Those connective tissue coverings, to review, are endoneurium, which is a loose connective tissue that encloses the axon and their myelin sheaths, including the Schwann cells. The perineurium, which is going to hold the bundles of fascicles together, and the outer, tougher fibrous sheath called the epineurium around the nerve organ itself. A very nice image of them here in 13.4a. And an artist drawing of what you see there in 13.4b. Most nerves are mixed or with afferent and efferent fibers carrying information in both directions, as well as both somatic and autonomic fibers within them. So we can classify nerves according to the direction of the impulses they transmit. So a mixed nerve would have both sensory and motor fibers carrying information to and from the central nervous system. Sensory nerves carry information towards the central nervous system, while motor nerves carry information away, afferent and efferent. Pure sensory or pure motor nerves are rare. Most often nerves are mixed. Types of fibers in mixed nerves would include somatic afferent, somatic efferent, visceral afferent, and visceral efferent, depending on whether it goes from muscles to brain, brain to muscles, organ to brain, organ to brain to organs. Ganglia are cell bodies that are associated with nerves in the peripheral nervous system. The ganglia are associated with afferent nerves. They contain cell bodies of sensory neurons. That would include dorsal root ganglia, which are sensory somatic. Ganglia associated with the efferent nerve fibers contain autonomic motor neurons. They would be the autonomic ganglia, which we will cover in the next chapter. Most neurons are amitotic. They do not divide. But some neurons, if the cell body is intact, the soma, the peripheral axon can be able to regenerate in the peripheral nervous system. This has not been shown in the central nervous system. And it is a very slow process in the peripheral nervous system. So central nervous system axons, most of the central nervous system's fibers never regenerate. The central nervous system oligodendrocytes bear growth inhibiting proteins that prevent central nervous system fibers from regenerating. Astrocyte at the injury site can form scar tissue. They can be treated with perhaps neutralizing growth hormones, blocking receptors for inhibitory proteins, and destroying scar tissue components. We really don't have the capabilities yet. The peripheral nervous system axons, however, can be regenerated if the damage is not too severe. Axon fragments and myelin sheath distal to the energy will degenerate. This is known as Wallerian degeneration. That degeneration will spread down the axon. At that point, macrophages come in and clean the dead axon debris. Schwann cells would be stimulated to divide, and the axon filaments would grow through that regeneration tube. As the axon regenerates, the new myelin sheath would form. And figure 13.5 walks you through those steps. And that is the end of our first section.